Hello students. In the previous lesson we have already learned about pollination. Today we are going to learn about fertilization in the flowering plants. Now pollination is basically the beginning, or rather we can say the first step of pollen pistil interaction. Now this pistil is the female reproductive part which consists of ovary, style and stigma. And the pollen pistil interaction basically begins with pollination and it is followed by the pollen adhesion to the stigma. Now this pollen which you can see here is a two-celled stage containing a tube cell and a generative cell. Okay. Now as the pollen lands on the stigma here, stigma secretes some substances which mainly consist of sugar and water, and this stimulates the pollen to germinate. Now as you know the structure of the pollen grain, it has got two parts, outer exine and the inner intine. Now in between the exine there is some discontinuous delicate areas which are called germ pore. Now from here the substance that is released by the stigma is absorbed and eventually the pollen tube starts forming. Now the length of the pollen tube depends upon the type of style. If the style is hollow type, then the pollen tube enters on the surface of the canal cells. But if the style is a solid type, then the pollen tube will enter through the connective tissue present in the style. Now, the pollen tube gradually makes entry to the ovule. Most commonly, the type of ovule we get to see is a anatropous ovule. We have already learned about the types of ovule. Now, the pollen tube then enters into the embryo sac. And this entry can be of three types. If the pollen tube enters through the chalazal region, then it is said to be chalazogamy. If the pollen tube enters through integument here, then it is said to be mesogamy. And if the pollen tube enters through the micropyle, then it is said to be porogamy. Now the most common entry of the pollen tube is the porogamy. Now as the pollen tube enters through the micropyle, synergids degenerate along with their walls. Now pollen tube, when it is making entry through the micropyle, first and foremost it releases male gametes, two male gametes. Now one male gamete fuse with the egg, resulting in the formation of zygote, a diploid structure, which is known as, the process is known as fertilization. And this was discovered by scientist Strasburger. The other male gamete that is released, it fuses with the two polar nuclei here and forms the primary endosperm nucleus. And this is termed as triple fusion. So, the primary endosperm nucleus give rise to endosperm, while the zygote develops into embryo. Now, developing embryo will get nourishment from the endosperm. Now, thus, these two processes together is called double fertilization, and this was discovered by the scientist Navaschin. And the experiment was done in Lilium and Fritillaria species. Now coming to the post-fertilization changes. Basically, antipodal cells and the synergids degenerate after their functions. Now, zygote will give rise to embryo, ovary into fruit, ovule into seed. The primary endosperm nucleus will change to endosperm, that is the nutritive tissue. By the time all changes takes place, simultaneously other changes are also observed, like the complete nucellus is used up. Now, when in some cases, sometimes some nucellus tissue still remains. So, if the partial nucleus with the seed is present or is observed, then it is said to be perispermic seed, like that we see in the seeds of beetroot and the black pepper. Now, another important change that takes place post fertilization is the formation of endosperm. Okay. Now, based on its formation, it can be classified into three types. The first one is the cellular type. Now, in the cellular endosperm, here the division is normal. That is, karyokinesis is followed by cytokinesis. Now, since it undergoes the mitotic divisions, the mass of cells that are formed are all triploid. This is basically found in coconut, that is the white edible part of coconut. Now, before we describe the halobial type, we're going to know about a type. Now, here the primary endosperm cell nucleus undergoes karyokinesis only. That means no cytokinesis occur here. This type of formation of endosperm is a nuclear type endosperm, and it is basically found in coconut water. The tender coconut that we get is the endosperm. So, basically coconut is formed by the triploid cell which undergoes nuclear division forming the coconut water, and the peripheral part undergoes cytokinesis forming the wall. Now coming to the third type, that is the halobial type, which is seen mostly in monocots. It is the intermediate type between the nuclear and the cellular types. So what we learned is that endosperm provides nourishment to the developing embryo. In most of the cases, it gets used up by the time the embryo develops. Now, this type of seeds where no endosperm is present are called non-endospermic seeds. But if the endosperm persists after embryo formation taking place, then it is said to be endospermic seed. We are going to learn about this in detail after the embryo development taking place, where it will further be classified into monocots and dicots. This will be taught in the upcoming lessons. Thank you.